A single gene can indeed have multiple effects on an organism, a phenomenon known as pleiotropy. In the case of pea plants, Pisum sativum, one gene affects both the size of starch grains and the shape of the seeds. To understand how this works, let's consider a monohybrid cross focusing on these traits. The gene controlling starch synthesis has two alleles, B dominant and B recessive. In BB homozygotes, starch synthesis is highly efficient, resulting in large starch grains, which also leads to round seeds after maturation. Conversely, BB homozygotes synthesize starch less efficiently, producing smaller grains and ultimately, wrinkled seeds. Heterozygotes B-flat typically display round seeds as well, suggesting that B is the dominant allele regarding seed shape. However, this pattern introduces a fascinating deviation from Mendel's law of dominance when we consider the size of starch grains. In heterozygous B-flat seeds, the starch grains are of intermediate size. This intermediate phenotype is a classic example of incomplete dominance. Unlike the complete dominance observed in seed shape, where heterozygotes appear identical to one of the homozygotes BB, incomplete dominance in starch grain size results in a phenotype that is a blend of both parental traits. Therefore, while the trait of seed shape follows Mendel's law of dominance with B being dominant over B, the trait of starch grain size does not. It instead demonstrates incomplete dominance, where neither allele is completely dominant or recessive, enriching our understanding of genetic inheritance patterns.